The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, welcome to episode 42 of the eLife podcast, a special celebratory edition of the programme recognising the 5th birthday of eLife. I'm Chris Smith. This month we're talking to two of the editors who make eLife tick to find out why they signed up in the first place and whether the journal has met their expectations. We hear from the person that made peer review accountable for the first time, and we look back at two of the earliest papers published in eLife to find out where their authors are now. In one case, she is watching bodies decompose in a desert, and in the other, he has hatched a revolutionary understanding of the evolution of the process that's keeping us all alive. But first, let's hear from Deputy Editor Dietler Fagel on the opportunity that he saw in joining eLife.
This really comes from my own experience over the last 30 years, how much more difficult it had gotten, especially for young researchers, to get published at the very highest level. When I started out, when I got my PhD in '88, it was essentially guaranteed when you did very good work that would be published at a very visible level. And that has really dramatically changed over the last 30 years.
In what way?
The scientific enterprise has grown enormously. Unfortunately, the outlets that are seen as being at the highest level have not kept the pace. They have grown a little bit, but certainly not at the same rate. What that has led to, you very often come across articles where this would not have looked out of place in one of the very general, broad interest journals. Conversely, it's still true that when you look at what some call elite journals, the work that is being published there is, of course, very good. But then you also realize that there could be probably 5 or 10 times as many articles that would be fit to appear there.
And how do you see what you've been striving to do with eLife as addressing that need?
Yeah, so certainly not having any arbitrary limits very much helps. It's difficult for me to know whether some of our competitors artificially restrict the number of papers that they publish every week or every month. It would seem so because they haven't grown at the same rate as the scientific enterprise has grown over the last 30 years. One should not turn away manuscripts that are at the same level as other manuscripts that one publishes. Very regularly, students or postdocs will show up and say, why did the journal turn away my paper? It's not any different really or worse than this other paper published here. They have the impression that what's turned away is inferior compared to what does get published at the very highest level. And that really is not the case. It's often really the luck of the draw. And this is something where at eLife we want to get away from. We strive to give much clearer guidance what is expected, and that we do not string authors along or we tell them, well, maybe at some time in the future we'd be more interested in publishing your work. And why don't you just try to add more stuff and convince us then? That's really completely contrary to the eLife ethos.
Yes, because some PhD students I have spoken to have spoken of their disenchantment when they have completed 3 years of work on a thesis, submitted a paper, and then had back from the reviewers about another 3 PhDs worth of additional experiments just to prove a point. Is that what you're getting at? You're saying, we're going to give you a direct answer, we're going to give you a quick direct answer, and if you fulfill those criteria, there's no holds barred after that towards getting your research actually out there.
Absolutely. The feedback that we provide after the reviews are much clearer, I would say, than what you receive from many other journals. For us, this is one of the most important things, to be very, very clear what we expect in a revision. We normally list a couple of experiments if we think more experimental evidence is being done. And if it comes back, the work has been done, then we very likely will accept it. No moving targets.
And finally, what do you think is on the horizon, or what's next on the agenda? What are eLife going to be pushing to do in the next 5 years to carry on being adventurous, daring, and trying to change the way that academic publishing is done?
Well, something that is very high on my list of priorities is actually after having demonstrated at eLife that our process works, to convince and, if you will, convert as many other journals as possible to take a similar approach, so that this really becomes the standard, and that in future students and postdocs will say, if there's not a transparent, consultative process among reviewers, I will not agree to submitting my manuscript to such a venue, no matter how fancy that journal is.
Dietler Flegel, who's also the director of the Max Planck Institute for Developmental Biology. Back in 2013, I spoke for the first time with Jessica Metcalf, an early career researcher who'd just published a paper in eLife looking at whether the bacteria recoverable from the body of a decomposing mouse can be used forensically to predict the time since death. Since then, she's been appointed as an assistant professor at Colorado State University, and she's moved on from mice to men.
When we spoke last time, 4 years ago, I had just published my first paper looking at how repeatable the microbial succession during decomposition was, where we decomposed mice all of the same age, in the same lab, on the same soil type, and we sampled them during 8 time points, and we found really it's the exact same set of microbes at each time point for each mouse.
Why were you doing that?
This was our first step in trying to understand whether microbial succession during decomposition is a repeatable enough and a predictable enough process to potentially be developed as a forensic tool to estimate how long a person had been dead. And that was the first paper that we published in eLife.
Where have you taken the research since? It's obvious that things have gone in the right direction for you because you've achieved a significant university appointment and begun to publish more on this topic. So how are you developing that initial eLife publication?
Right, so our first publication in eLife really showed us that if we control all variables, that we see this really repeatable succession of microbes during decomposition. And so then this launched a number of other projects where we start focusing on different variables and start changing them and ask the question, do we still see this repeatable succession of microbes during decomposition, which is what we need if we're going to be able to develop this into a forensic tool. In 2016, we published a paper in Science where we described 2 big sets of experiments. In the first one, we did a very similar mouse study as we did in the eLife paper, but we used 3 different soil types that had really different starting microbial communities. And there, we were using soil as a proxy for environment. If a person dies in Moab, Utah, or in the mountains of Colorado, are they going to have the same succession of microbes, or is it going to be completely different? And this helps us understand, is there a universal clock that we can develop to estimate the time since death, or does it need to be regional? And what we found there is that really the soil type didn't matter. We still get a very similar succession of microbes. And then the second part of that paper, we actually were able to work with Anthropological Research Facility down in Sam Houston State University, where people donate their bodies to become part of a forensic science experiment after death. And so we had the opportunity to decompose 4 human bodies, 2 in the spring and 2 in the winter. We sampled the skin and the soil over time. And we again asked the question, in this outdoor scenario where there's rainfall events, there's insects, there's scavengers, do we still get a repeatable succession of microbes during decomposition? And what we found was yes, we do, and it's fairly impressive. That was a real shock because, we went from a lab setting where we were controlling so many things to this outdoor setting, and we were still able to recover this pattern.
So what do? Do you just lay the corpse on the floor as though the person had died in situ? And are they clothed, unclothed? Because that could make a difference too, couldn't it?
For the studies that we've done so far and that we have currently going right now, we've placed all the bodies in the exact same way. So we're still controlling for that variable at the moment, but at some point we'll probably test that as well. Everybody's outside, laid face up without clothes.
Now, why do you think that you see these results? Because the soil is such a strong driver normally for what's around and what it can support and sustain. Is it that the body is so nutrient rich and so replete with its own microbial spectrum that it just basically feeds and outcompetes anything that could come in from the environment, and that's why you see this very reproducible succession of microbes?
What we found is that on average, most microbes appear to actually be coming from the soil. So you have to remember that soils have an incredible diversity of microbes, and even though two soil samples may look very different overall as far as their microbial diversity, they still have some of the same microbes. And so what I think is probably going on is that these are just really ubiquitous bacteria that are at a low abundance at a lot of places across Earth, and they are very good at responding quickly to a new nutrient source. The body is this huge pulse of nutrients, and there are microorganisms that have evolved to be really good at, once they find that, boom, they're growing like bonkers.
Jess Metcalf. You're listening to the eLife podcast with me, Chris Smith, and in this episode we're celebrating 5 years since eLife first opened its electronic doors to submissions. Still to come, how photosynthesis has evolved and why one editor says that eLife offers a scientific publishing route that goes back to the future. But before that, peer review is something that we all take for granted. We wouldn't dream of publishing something credible these days without it. But the system wasn't always robust. And someone who's really helped to shake things up over the last 30 years, founding in the process an internationally recognised conference series called the Peer Review Congress, is former medical doctor and high-altitude physiologist Drummond Rennie.
What happened was that I was working in a huge cardiac clinic in Guy's Hospital in the early 60s, and I got interested in the problems of blue babies, kids with congenital defects in the heart, and they couldn't have their blood fully oxygenated. Now, you get blue at high altitude, and I did all the research I could do in London. I moved to the US in 1967. The research I did then went very well indeed. This was high altitude in the Andes and the Himalayas and Alaska and the Yukon and so on. And an extraordinary thing happened to me. I'd finished some studies in the mid-1970s, and I had what I consider in retrospect the luck to fall a longish way, actually a very long way, down a mountain in the Canadian Yukon. From being a really second-rate climber, I became a fourth-rate one, and could no longer take full responsibility for experimental people who'd agreed to be experimented on that I'd taken to high altitudes. And the lucky part of that was that I was absolutely forced to change my career. I'd probably run out of ideas anyhow. It stopped me from getting killed later. And I became the first deputy editor of the New England Journal. Looking at what I had to handle and deal with, absolute abysmal standards, the sheer weight of rubbish. It was astounding to me. This stuff would flow in and we'd flow it back again. The good research was superb, but I'd say a third of the research sent to us were trite rubbish. One of the common features, it occurred to me, of these trite rubbish papers was that they'd all passed rigorous peer review. One way and another, I became deputy editor of JAMA, the Journal of the American Medical Association. At that time, it was in a bad state, and I was intrigued by the idea of helping to rescue it. I found that people there were interested in peer review also, but were prepared to go a little further. And in 1986, I wrote an editorial announcing that we would back a conference to discuss peer review. But I put in and insisted we would only present research. I was fed up with pooh-bah editors just saying, we've had this disaster with peer review, but our peer review system we declare to be excellent on the basis of no evidence whatsoever. So what a nice thing to gather evidence, and an even nicer thing to get clever people out there, if there were any, to find this evidence. And so in 1989, we held the first conference on this, and it was an amazing event. Angry arguments and a great deal of excitement. So we've been having them every 4 years since. The first thing that happened was the amount of rubbish being printed was confirmed. So these conferences during the 90s and the 2000s, early 2000s, gave a tremendous impetus to the idea that the literature was full of biases, and to the literature that said these are ways of correcting all of these biases, and then of helping journals follow guidelines that would enable evidence-based reporting. So that is what happened very broadly. The last thing I'd say is something a little different. There were one woman for every 10 men at the first Congress. That ratio has changed completely. My parents got married in Cambridge around 1933. Before that, my mother put herself through medical school in the 1920s. She'd have been very happy with that ratio, don't you think?
I'm sure she would indeed. Drummond Rennie there, the architect of the Peer Review Congress, the 8th meeting of which just took place in September in Chicago. Time to talk to another former eLife author now. Julian Hibbard is based in Cambridge, and he's been trying to unpick how the more advanced modern form of so-called C4 photosynthesis has evolved. We first met 4 years ago when Julian published evidence that C4 photosynthesis had probably evolved not once, but many times. And it's a story he's since been able to refine and build upon.
The ancestral form of photosynthesis is effectively unchanged since bacteria initially evolved the ability to fix carbon, so probably 3.6 billion years ago. The majority of land plants still use that system, and that system relies on an enzyme which sounds unfortunately like a breakfast cereal called Rubisco. Rubisco is great, it fixes CO2 and makes carbohydrates, but some of the time it makes a mistake, and instead of taking CO2, it takes oxygen. And because the concentration of oxygen in the atmosphere is now very high compared to that 3.6 billion years ago, those mistakes happen more often. It wastes energy, it generates a toxic compound. The C4 pathway is a system which effectively abolishes those mistakes by generating a little turbocharger around this Rubisco enzyme such that more CO2 is supplied to it.
So back in history then, the bacteria that evolved this in the first place were living in an environment which was dominated by carbon dioxide, hardly any oxygen around, so they didn't need an enzyme that really could discriminate between an oxygen molecule and a carbon dioxide molecule. And it's only in the latter era now, this is more of a problem, so there is this pressure to adopt this turbocharged form.
That's exactly right, yes. Over evolutionary time and geological time, the CO2 has come down because photosynthesis has been so successful, and we're now in a completely reverse situation where CO2 is relatively scarce and there's huge amounts of oxygen, and that's led to this conflict between fixation of oxygen by Rubisco and the fixation of CO2.
How has it changed in order to make it a better discriminator between an oxygen molecule and a carbon dioxide molecule?
So in the C4 pathway, plants have developed a complex suite of traits which allow CO2 to be pumped from one cell type to another cell type, and in that second cell type we have the Rubisco enzyme, and that increases the concentration of CO2 specifically around Rubisco, which basically abolishes then the oxygenation reaction.
So it's like a sieve?
It is a bit like a sieve, yeah. So that sieve involves a specific set of enzymes which initially fix the CO2 into a different form, The CO2 is then released at high concentrations around rubisco, and that's what's improving the efficiency of photosynthesis.
And if I look at all plants that do this, have they all got one common ancestor then? Or does it happen with the same common end product, you get lots of CO2 around your rubisco, but the way you get there is different in all these different plants?
There are multiple ancestors of C4 plants, so we estimate it's evolved at least 60 times. And we think there's a general set of machinery in C3 plants which is sort of nascent, and it allows C4 photosynthesis to develop. So a lot of the regulatory structure associated with turning genes on or turning genes off, we have discovered more recently that quite a lot of that is sitting there in the C3 situation. It's not being used exactly as it would be in a C4 leaf, but in a way it's ready to be used.
And so because it had that machinery, it's a bit like me walking into a kitchen and saying, well, that's a knife and fork. We will usually use that for eating, but actually I'm going to beat an egg with that. And so you're using a fork which is already made as a fork, but you can do a different job with it, and it's useful for both.
Yes, and I think that's a great analogy. And actually, some of the work we've done more recently is a little bit more nuanced than that. Maybe it's like there's a fork sitting there and we would normally use it for our dessert. We're going to carry on using it as a fork, but maybe for a different course. Sounds terribly Cambridge, doesn't it? But if we're thinking about these forks and we say, well, actually, this is a gene for a fork. In the ancestral C3 plant, those genes are very poorly expressed. In the C4 pathway, they're very highly expressed. And to our surprise, what we found is that in the ancestral C3 state, a lot of those genes are already regulated by cues which turn on photosynthetic genes, so such as light. They're actually sitting there as part of that photosynthetic architecture, regulatory architecture for photosynthesis, but they're not responding to it fully. So all you need to do is amplify that response as you go from C3 to C4, and hey presto, you have the full system working.
And is your ultimate goal then to understand photosynthesis, understand how plants work, so that we can do it even better?
That's one of the potential outcomes. There's a broad consensus now that despite millions or billions of years of evolution of photosynthesis, it's not optimized for current agricultural practice. There's a broad consensus that there are multiple ways of improving photosynthesis. And so if we understood it enough to be able to engineer it into a C3 crop, such as rice or wheat, we should be able to dramatically improve crop yield, so up to 50% improvement.
Julian Hibbard. And we finish now by hearing another perspective of what's been achieved so far in eLife's history from another of the journal's deputy editors, who wants to stress why, in her view, eLife is a move back to the future.
My name is Eve Martyr, and I'm a professor in the biology department and neuroscience program at Brandeis University. When I was a young scientist, papers were intended to be complete and full descriptions of a piece of work, and to lay out the data, lay out the controls, lay out the methods, and then to allow the authors to articulate what they thought the work meant. And over the years, a lot of that became lost as people pushed for publications of higher and higher impact to the detriment of clarity, thoroughness, and to my mind, eLife is a way of going not back to the past, but forward to the future. We can write papers the way they should be written.
And do you think that's happening?
I think that's happening, not perfectly. Many of the papers we publish are papers that I'm very proud of, papers that have enough depth, enough scope, enough clarity that they allow the reader to really see what the important findings are and how the work was done.
How is eLife competing with other journals, though, that still do subscribe to what science publishing had become, with people submitting papers and then being given another 5 PhDs worth of experiments to do before they get their paper revisited?
Many eLife papers are quite substantial, so it's not that other journals are publishing more substantial papers. But eLife refuses to send authors back for 6 or 9 months or 2 years of work. We basically follow the policy that authors should send the paper when they consider it done. And then we have tried to evaluate it as it is. And so we might ask for minor modifications or a small bit of additional work if, for example, a reviewer says that a specific figure has too much noise in the recording, or someone thinks that a Western blot is fuzzy. We might ask them to redo something like that. But we will not provisionally accept a paper and ask them for 9 months worth of work, because we think it's really unfair to the young scientists who are doing the work to stretch out their careers and leave them in scientific limbo for a very long time.
How are you doing that, though, without potentially compromising on the quality?
It's very simple. I'd like to tell you a little anecdote that tells you the difference between eLife and what's happened to a lot of other publishing. Very early on in eLife, we sent a paper to a young man who's a very fine scientist. He was a beginning assistant professor, and he reviewed the paper, and he listed 6 or 8 experiments that should be done, probably a year's worth of work. And in the consultation session, I pointed out to him that we were trying to avoid doing that. So I asked him whether those experiments were necessary to support the point of the paper. And he said, "Oh, no. I just thought the job of a reviewer was always to suggest more experiments." And so what we do, if experiments are really necessary to prove the point of the paper and they would take 6 months, we reject the paper. And we tell the authors that they are free to take the paper elsewhere. They're free to do the experiments and maybe resubmit it to us or elsewhere. But basically, we don't play those sorts of games with authors.
When you say to colleagues who are suggesting to you that they might have some work they want to publish, and you say, what about eLife? Have you noticed that people react differently now compared with, say, 5 years ago when things were just getting going?
Oh, absolutely. I've noticed something else which is really important. Now when I go to seminars or a conference, oftentimes you see people showing slides where the work was from an eLife paper. Some very, very prestigious scientists now are routinely citing work that they've published in eLife. So I feel like we've really crossed a very important threshold.
Where are you going next? What's going to be the sort of strategic direction for the next 5 years? You've honed down the peer review process. That seems to be bearing fruit. You're attracting some very high caliber publications. You always have, but the numbers are definitely going up. What are you targeting next?
We're hoping to grow in certain areas. eLife has been more successful at attracting the best papers in subfields of biology than in others. I feel like we've grown eLife tremendously in the past 5 years, and that we've done it gradually enough so that we haven't lost the essential eLife ethos.
Eve Marder. And that's it for this episode. You've been listening to the eLife podcast, previous editions of which are also on eLife's website, that's elifesciences.org, or you can subscribe via iTunes. This is a Naked Scientist production. I'm Chris Smith, and you can find out more about what we do on our website at nakedscientist.com. I shall be back next month to take a look at how monkeys armed with tools are wiping out marine life and why science papers are getting harder to read. But not in eLife, of course. Until then, thanks for listening and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.